Lots of terrific information, so if you could please start formulating some questions for the end of the session after everybody has presented. One of the things that I wanted to bring up and talk about real quickly while we transition here is that as science grows and technology begins to increase and get more complex, that there's a call for interdisciplinary collaborations between different science communities. So in the past, biologists didn't have the tendency to talk so much to the chemists or the bacteriologists. But now, during this age and age, with the increasing amount of technology and information that's out there, these collaborations have to be a part of the everyday science in order to move science and our understanding of science forward. And this actually was talked about a little bit earlier, about this increase of this new type of science called translational science, or from the bench to bedside science, where basic research findings are now being translated into therapies or treatments for persons who are afflicted with this disease that's undergoing research. And so there's a collaboration between the research scientists and the physicians, sometimes like our next speaker, who the physicians are trained as scientists, cut out the middlemen, and talk about some really great things. So next I want to introduce Dr. Jeffrey Henderson. He's Lakota, and he's an enrolled member of the Cheyenne River Sioux tribe. In 1998, Dr. Henderson founded the Black Hills Center for American Indian Health, a community-based research organization where he spent much of his career examining the numerous prominent American Indian health inequities, and then using highly participatory methods in determining how to best address them through asset-based approaches. And he currently serves as an assistant professor at the University of Colorado, Denver's Native Elder Research Center, and is the author of 4 books and 4 book chapters and over 40 scientific manuscripts, including In Their Own Voices, American Indian Decisions to Participate in Health Research, published in the Handbook of Ethical Research and Ethnocultural Populations and Communities. So with no further ado, let's bring up Dr. Henderson. This banner is getting me here. It won't go away.
Hihane washte me tako yepi. Jeff Henderson na wawokia imachiapalo. Good morning, my relatives. My name is Jeff Henderson. My traditional Lakota name, which is a Lakota person I'm encouraged to share with gatherings like this, is Wawokia, which means the helper or the one who helps. Not all Native peoples can share their traditional names with such a group, though, so I don't want you to go away thinking that every Native person you meet, you try to wring their traditional name out of them. I really appreciate my relative Francine's presentation. That was a really great presentation, and it helps to set aside those things in order to simply answer the question, can genetic research improve Native health? Yes, it certainly can. Has it already? Yes, it certainly has. And so let's take a look at a few of those. There's been described in the literature a constellation of Southwest Athabascan genetic diseases now. There's severe combined immunodeficiency syndrome, there's Navajo neuropathy, Navajo poikiloderma, also Athabascan brainstem dysgenesis. All have been tied to discrete, in most cases, single point mutations in the genome that are felt to have derived from at least one and possibly several different genetic bottleneck events that happened in the Athabascan people's history. Certainly, pharmacogenomics have helped our Native people who have been patients in cancer centers. It's helped to define their rates of metabolism, a variety of chemotherapeutic agents now that have come about and are being used regularly in clinical care. You might be interested to find out, too, that we now know, for the most part, that patients who undergo radiation therapy for tumors, their tendency to suffer, in some cases, very significant skin burn is under genetic control. And in fact, we've done, not we, but the group at Rapid City Regional Hospital, led by Dr. Dan Peterwright, has characterized the genetic alleles that are responsible for that radiation-induced skin injury among a cohort of Lakota patients from western South Dakota. So certainly it's the case that genetic research has helped, and most of these examples are in a patient care vein. There's also, you may be well aware of the so-called Lynch syndrome, or hereditary non-polyposis colorectal cancer. Dr. Lynch gathered up all the seminal data that helped to identify this particular genetic disorder that is now known to account for 3 to 5% of the 178,000 or so colorectal cancers that develop in the United States each year. He developed that data in a cohort of Navajo patients on Navajo Nation, and so the benefits of that particular research have spilled over to the general population. There's more research going on. 3 to 5% is getting close to being able to establish the ability of such markers to serve for carrier screening. It's not quite there yet, nor is it with that constellation of Southwest Athabascan genetic diseases, but with a little bit more contextualization, a little bit more time running forward in a prospective fashion, in both cases, we might have good future carrier screens in the population. So genetic research can be helpful. Broad population-based benefits are a little less clear, though. It's particularly the case that we know that tremendous amounts of money are being thrown at full genome scans or GWAS studies of complex trait disorders like type 2 diabetes, obesity, cardiovascular disease. Certainly, a huge amount of money is going for virtually no gain whatsoever, certainly among American Indians and Alaska Natives, and it's really doubtful that in our lifetimes, or certainly in my remaining lifetime, that we're actually going to realize much return on that massive investment that we're making in this particular field. So, what would be an example of genetic research that does have the promise to broadly improve the health of Native peoples? I just happen to be funded by the National Cancer Institute now in my 5th and final year of a study that is looking at both genetic and non-genetic factors related to smoking and nicotine metabolism among a large cohort of American Indians, both in the Northern Plains and in the Southwest. So let's take a look at that. This is to review for us. This is looking at dopamine and what happens biochemically in our brains with respect to dopamine. And I'm not going to step through everything here except to note that this is tyrosine hydroxylase, which is the rate-limiting step in the synthesis of dopamine. And exposure to nicotine and a variety of other substances can powerfully increase the basal rate of secretion of dopamine. And exactly how that happens, we're not exactly sure. It could be that it ramps up production and activity of tyrosine hydroxylase. It could make it so that dopamine sticks to its dopamine receptor across this synaptic cleft for a longer period of time. It could be that dopamine is not taken up by its transporter back from where it came, and it could be also that there's problems in breaking dopamine down and metabolizing it. All of these could be responsible for the reasons why the effect of dopamine is prolonged or lingers as it is, as we know it is in smokers. This is to simply point us out. This is showing the dopamine system where it courses throughout the brain in blue and the serotonin system in orange. And of course, we're familiar with many of the types of feelings that a smoker gets after they smoke a cigarette. It's also the case that nicotine itself, in addition to dopamine and serotonin getting metabolized, it's the case that nicotine gets metabolized in our liver by our cytochrome P450 system, by CYP2A6 gene specifically, and that gene works to break nicotine down to cotinine and cotinine further down to 3-hydroxycotinine, so it gets excreted principally in our urine. we now know, and a lot of work has been done in populations around the world to highlight for us, there are over 40 different variations of the CYP2A6 gene that have been described now, and we're just starting to look at the performance or the allelic frequencies of this gene in native populations right now, including in my current study. This slide shows us the effects of drugs on dopamine release. You may have heard, many of us have heard, that nicotine that you get from smoking a commercial cigarette bumps your dopamine level up about as much as heroin does, or morphine, and that's true. You can see that they're pretty comparable right here. Cocaine's a little bit better, maybe about half again as much. Amphetamine, though, is 4 to 5-fold greater in the basal release of dopamine in response to being exposed to amphetamine. Now, crack cocaine or crystal methamphetamine remarkably is up around 5,000 or so. A massive, massive release. And so, some of us are interested in what crystal meth is doing to our relatives, and including on tribal populations around America now. Well, there is a massive boost in dopamine that is why a person does that and why they're likely to become dependent on that particular feeling. We've just completed recruitment for this particular study. 526 of my Lakota relatives in western South Dakota and 210 individuals in the southwest around the Phoenix metropolitan area from a variety of different tribal backgrounds, in fact, not any one specific. This is a look at the model that we developed for this particular study. Such a study does not happen without the participation of a lot of different project team members, including vital tribal field data collection partners at MBERI. In this case, is a unique tribally based field research contract organization that stands for Missouri Brakes Industries Research, Inc., and they are wonderful partners on lots of different kinds of studies, not just this one. The central lab for this study is at Penn Medical Labs, and our 2 genotyping centers, so all dopaminergic and serotonergic systems for my study are being typed at University of Texas MD Anderson Cancer Center in Houston, while the CYP2A6 was discovered in Toronto by Dr. Rachel Tyndale, and I believe it's still true that that Toronto lab is the only place, certainly in North America, that you can get the CYP2A6 gene typed. And so the University of Toronto is involved in that particular study as well. So we have finished with, the Tyndale lab is finished with their characterization of the CYP2A6 gene among most all of the South Dakota sample. They've yet to take a look at the Arizona sample, though. And just simply among my relatives, among the Lakota, their lab is reporting out the highest rate of metabolic activity of the CYP2A6 gene that's ever been described so far worldwide. What does this mean? So it means that we break down in our livers, if our liver's functioning properly, we break down nicotine to cotinine more rapidly than has ever been described before. And the consequence of this, though, is that such individuals experience greater nicotine dependence. They also tend to smoke more than folks who have lower metabolic activity of this particular gene, and they have more difficulty quitting. I served for 6 years as a primary care internist in the Indian Health Service and was clinical director on my reservation at Cheyenne River, and then also head of mental health and social services at the Sioux Indian Health Service Hospital, a former tuberculosis sanatorium in Rapid City through the mid-90s. In the early 90s, when I was at Eagle Butte, we just finally got access to topical nicotine replacement patches, and they were originally produced and issued in 21 milligram sizes, and so we were following the guidelines that we were given and putting them on our relatives, and finding that that our relatives were getting extraordinarily sick, nauseous, and vomiting in response to the initial size. So we started instructing them to cut those patches in half. And even still, over half of our patients were getting very ill on that patch. And so we had them cut them again in half. So they were taking a quarter of the original 21 milligram nicotine replacement patches to get a therapeutic benefit from that. Of course, now the patches came to be produced in a variety of different sizes and dosages. But that fits into why we're doing this particular type of a study. What this study is going to do for us is show us, for the first time, it's going to give us hard evidence on why certain medications may work versus others for nicotine cessation. It's going to help to define for us and give us additional, hopefully more impactful information with which to pursue prevention messaging among youth, to try to keep our youth from starting to smoke. It should also certainly point the way to future intervention research in the cessation realm in a much more highly targeted way than we've ever had before. So hopefully I'm not overselling the promise of a study like this, which is a candidate gene approach to genetic research, but I think that hopefully you'd agree that yes, this is the kind of a study that potentially could have pretty broad population-based impacts. I'll leave it to others to say a little bit more about the adverse effects that have happened with the handling of DNA and our relatives' DNA over the past. Certainly there have, and I've suspended sort of my skeptic's hat for this, save for just a small little comments. Vince, I really appreciated your talking about the ethical, legal, and social implications. I've been involved in 2 of the projects from Morris Foster and Paul Spicer that they've worked on over the years, and certainly there's still more work that needs to be done in the LC arena with respect to genetic research in Native peoples. However, I will say that I think it's entirely the case that properly designed studies should be able to derive benefit to Native peoples and should be able to do so in our lifetimes. So with that, I thank you very much and I'll look forward to questions at the end.
Excellent. Thank you, Dr. Henderson.